Hello, everybody. Welcome to an ad hoc COVID-19 episode of 26.1 AI podcast. Brian Ray and I today will speak with Brett Averso, a founder and CTO of Evolve, a startup working on coronavirus.
Welcome, Brett.
Hi, thank you for having me.
Brett, you have some big news that's pretty recent in terms of what Evolve and what you are doing with immunoprecise antibodies. Do you want to start out with that?
Yeah, for sure. We have a collaboration agreement in place with ImmunoPrecise Antibodies, which is just a fantastic Canadian-based company that has expertise in developing therapeutic monoclonal antibodies. They have several very innovative platforms for developing antibodies in the lab. And what's cool is we have this great opportunity to develop antibody sequences from the computer, which is a process we call an in silico discovery process, and send those sequences to our partner, who will then in turn be able to produce those in a recombinant setting, and then validate and test those antibodies. So the AI is another complementary platform for our partner. Yeah.
And so how is this process different from, given the urgency and everything else that's going on right now, how is it different from the work you've done in the past?
Yeah, sure. So, the conventional means of discovering a therapeutic, right, involves countless of hours and days, and if not weeks, in the wet lab, trying to express enough antibodies or proteins when it comes to biologic therapeutics, in order to test those antibodies for their binding affinity, and some of them may be immunogenic, some of them may not be, some of them may be a therapeutic. They're hitting a target that's called an epitope that could elicit a response potentially, some could not. This takes a very long time. I think the really nice benefit of what we're providing from our side is a computational approach that models aspects such as the stability of the antibody, or the binding affinity, or a lot of these characteristics. And we're doing this all within the computer, so we reduce a lot of the time, the cost, and the upfront risks that could be associated with the lab development. And hopefully, as our technology gets more proven through our collaboration, that we will have found some greater efficiency in the drug discovery process. And yeah.
Well, I think I read that you're going to run through something like 10 to 150th power, the number of experiments that
Yes.
you're going to run in silico.
Right. Yeah. So the theoretical search space could be as high as 10 to the 150th. If you were to just strictly search for sequences from a combinatorics perspective, so think almost like a brute force means. But what we do here is we look at the relationships between the various amino acids that comprise of the antibody, and understand their positions and their relationships to one another. And we can kind of infer a coupling strength. So like a pair is, in a sense, like a coupling, and we can infer like a energy landscape across the whole biological sequence. And once we've inferred that energy landscape, we're able to kind of create like an inferential machine that has strong correlation with evolution and homologous or other similar antibodies that exist. So we have a smarter mutagenic type engine to develop antibody variations that could potentially bind better, could potentially perform better with respect to their stability, possess better traits, and possess better affinity for this latest version of the SARS coronavirus.
Yeah. So when what it really you're saying for in layman's terms is that it's going to help reduce the limit the effects of the disease. Is that correct?
Yeah, I think what I'm trying to say is that uh, we know that like today as we speak, there are antibodies that bind with the original SARS virus from the 2003 SARS outbreak. And right now, those same antibodies do not possess the same affinity for this latest strain. But if we can run these through our computer models, we may be able to land upon one that has great cross reactivity and the right traits to be a therapeutic. And that's what we're aiming for. I
And to get
in that way.
Sure, and to give a scale of the time savings, maybe explore that a little bit. Just for an example, I'm familiar with Zymergen and what they do with trying to predict useful microbes.
Sure.
And the example that Janet Matson, one of their ML practitioners, had given was 10 to the 17th number of variations in gene edits could take in traditional wet lab settings 10 to the 10th power of time, which is the age of the universe.
Mm. Yeah. Yeah.
So I imagine you're working in silico in kind of the same scale of time savings compared to traditional
Yeah.
lab setting.
That's an interesting way to compare it, and I think I think that in terms of actual drug development time, right, where it takes weeks for you for say if it was they have a hybridoma or B cell platform, and then another platform is called phage display. Either way, with these platforms, right, they're fantastic platforms at expressing antibodies towards a particular target, but it does take a long time. I think that when we're able to sample and generate sequences and filter them down all within the computer, it's a matter of days as opposed to weeks, and that's part of the asset of AI, and also gaining an understanding of what those potential candidates could perform like in the lab with our models, is the other major asset to what's going on. So
And
as
so,
yeah, sure, go ahead. Yeah, go ahead.
no, you and Andrew, you're spending like 100 hours in the lab or 100 hours working together a week. I heard. Is that true?
we're working pretty hard. We're working pretty hard. I would think like between like 80 and 100 hours a week is about right.
And I believe you're doing good. Can you talk a little bit more about, since we have some practitioners that listen to us, can you talk a little bit more about what your lab settings like? How do you apply this AI and machine learning to your problem? Is it in the cloud? Is it on premise? Are you using GPUs? That sort of thing.
Sure. Yeah. It's all on the cloud. It's we work primarily with AWS. We also have a few instances with Azure. We work with we do use a deep learning instance for a deep learning algorithm, but the deep learning algorithm is more under development right now. Right now, we're very focused on when it comes to our search space. We're very interested in graphical models, couplings analysis, and performing principal component analysis and various means of feature engineering on our filter side. So what we do is we generate a number of sequences at first. We generate several million sequences at first, and then we need to filter them out. So that on the filtering side, we characterize all the sequences and compare them to therapeutics that have been approved, and we work from there to try to come up with a great panel of antibody candidates that we think may bind.
So it's a unsupervised exercise. So it's basically you generate a bunch, then you use unsupervised learning PCA to narrow it down a bit.
Yeah, you know what? And let me clarify a little bit, because I think my mind just jumped around to PCA for a moment because I was actually working on something like that. But we work with different algorithms. There's a lot of features to characterize the proteins by, but we apply like a PCA for some of the reducing of the features, and then we have a different classification methods that we use to classify the antibodies. We cluster them based on their similarity, and then we'll cluster them based on the characteristics they possess. So we can cluster them based on, say, their overall thermostability or their overall hydrophobicity versus charge. We have a whole bunch of host of different features. We try to explore our data set to get a very diverse set of variants. And then lastly, we apply a Markov process for modeling binding affinity.
Markup chains, you mean? Yeah, Markup chains.
Yeah, Markov chains, pardon me. Yes.
In terms of your team, what's that look like, since you're not a biologist, right?
I'm not a biologist, correct. Actually, it's interesting. So Andrew and I are both not biologists, and we kind of came at this as outsiders, thinking about it as data scientists, and asking ourselves, how could we look at challenges differently. So we do have a great team of science guys with us, and they've been extremely helpful in us discovering and understanding as we go. But it's been probably the steepest learning curve in my life, is learning biology in this level, learning structural biology, or learning about like essentially like biophysics, understanding how these features and these characteristics describe proteins, and it's been a very, very interesting challenge and path for us so far.
So you're a data scientist at heart, and you're enjoying the rich feature space in biochemistry.
Absolutely. Yeah, absolutely. Yeah. The understanding this domain has been very challenging, but we have some really great guys on our team that have been extremely helpful and that we're very grateful for. It's been a lot of challenge. It's been a lot of fun. And I think that we're gonna keep being kind of like an unusual team. We're focused on a data science perspective to this, as opposed to kind of a conventional biotech perspective. So that may mean that our team is may not possess our technologists may not necessarily be bioinformaticists, for example. Our technologists may have a very heavy background in data science and deep learning and machine learning. I think that's something that we like to do, just to be a different company.
From stage of company for you and Evolve, you're pretty early, correct? And your relationship with IPA is pretty new, and in some sense they're part of your probably norming of your sales process, I'd imagine.
Oh yeah, absolutely. Our relationship with IPA is fairly new, and this is something new for us. I think we want to. I think our primary means today is to get as many sequences as we can that are diverse out to IPA, so they can produce and validate these sequences. We'd love to see how these sequences perform in the actual lab, so we can correlate that back with the results our algorithms are predicting, and that's going to help us gain greater insight as to where we need to improve our platform, and help essentially validate whether we're on the right track.
And you started out in finance too before Columbia, is that correct? And how does you go from, your interest in finance and this what appears to be extremely deep knowledge into machine learning applied to biology?
Yeah, it was a really big change. When I was in finance, it was, I didn't feel much intrinsic reward to what I was doing, and so when I moved into when I came to the decision to go into data science and then go down this path, I think it had to do a lot with how can I use information um, to make people's lives better, right? Um, and a lot of times in finance, it feels like I'm three or four orders removed from that kind of a process. I mean, I'm working with data every day, right? But here at least I'm sitting on the edge of like the potential outcomes from the models, could have a great impact on humanity, right? And I mean, not just with COVID-19, but with drug discovery in general, right? Given how challenging it is to get a therapeutic through up to even get to clinical trials is quite challenging, right? Most candidates will not. There's only one candidate out of a batch that could be chosen to go through the clinical trial process. So if we can maximize the possibilities and use models to infer and predict how they'll perform, I think that's an amazing benefit to society and to medicine.
And the race is on right now for therapies that are effective for coronavirus. Do you know of other folks approaching this problem in the same way or in a similar way as you and Evolve?
I'm sure there are quite a few companies that are also trying to leverage AI, either through collaborations or to some degree internally. I think that there's so many ways in which a therapeutic could fail in trials, and I think it's good that the whole industry is galvanized in trying to discover various therapeutics, whether it be a vaccine or a monoclonal antibody, or a small molecule being repurposed towards COVID-19. I think it's a very good thing that everyone is kind of working towards this and trying to move as fast as we can. But it's just going to take time, unfortunately, and time is not on our hands. So
The pressure's on. Yeah, the pressure's on. So
pressure's on.
update us. How is it looking? How are you? Do you have results, or how far are you?
How soon do we save Brian from his stir crazy life right now?
Yeah, can you get me out?
Being locked down.
Yeah, right. Yeah, I know how you feel. I feel very stir crazy now as well at this point. I think that so we've submitted a panel and we're reviewing the characteristics of that panel presently. That panel has quite a lot of features and characteristics about it. So it is kind of a lot to go through, but the way we're working on this is kind of iteratively. So our scientists are kind of getting together with us together, and we're talking about what are the most important features with respect to the selections that we want to make. We look at different clustering visualizations to decide not just on sequence diversity, but also on different performance characteristics. We try to cluster all the antibody clones in different methods because we want to provide a very diverse sample for the lab. And then lastly, we also want to think about with a sense of control for our experiment. So we're also thinking about, perhaps what are several antibodies that the algorithm predicted would perform only fairly, or and then kind of use those as a control, and that would also help validate our algorithm's predictive accuracy.
So in the subtle detail here that a lot of the other machine learning people who listen to our podcast might pick up on is that now you have to go and kind of collectively decide which features are important kind of after the fact, because there's no answer. There's no right answer yet, right? So you're now going back and you're looking at some of these features types and you're ranking those as well. Is that correct?
Yeah, that's right. A lot of it is about exploring the feature space, and a lot of it is also about what sequences do we wish to eliminate that of our candidates based on those features performances. And then also, as we gain insight through these conversations, we're able to develop new algorithms that could predict or recommend different sequences. Based on our conversations with the scientists at IPA. So it's all very very informative for us. So we're very happy for this. Having this collaboration is a really big opportunity for us, especially because we're so early stage right now. So.
And what you're looking for right now, is it a treatment instead of a vaccine? For example, Johnson and Johnson announced that they've selected something that's promising, for example, for a vaccine. Is that specifically what you're looking for, or is it more of a therapeutic molecule that would help people recover from COVID-19?
Yeah, well, that's the interesting thing about monoclonal antibodies, right? They're typically applied as a therapeutic, and they're particularly successful in a host of different diseases. But monoclonal antibodies could also be applied in a prophylactic setting. So like there is the possibility that you could be vaccinated with monoclonal antibodies and receive a temporary immunity. But generally, I think the application of our therapeutic would be as a therapeutic. Excuse me, that sounds silly. The application of our platform would most likely be as a therapeutic for those who have contracted COVID-19.
And to bring it up a layer higher, and I kind of switch gears here because I always like to ask this question. Now you're doing good things here with solving a problem for coronavirus, obviously. What's the flip side? And maybe even in the same domain, what if you get it wrong? Or what if the chances of mutation for the virus itself, or, I always try to look at both sides of the coin.
Right. Yeah, I understand what you're saying. Yeah, the risks are always very high, especially in drug discovery. Fortunately for us, when something fails, given that everything that we're working on is in silico, it's not too difficult for us to try to re-engineer and figure out how we can improve our algorithms, or if we should try another method or another means. It could just be that the target that we were going after isn't the right target. It could be that the target is not immunogenic. It could be that it could be a whole host of different reasons with respect to why something doesn't work out. I think that it's important to we try to keep an open mind and think about how we could pivot our tech stack if something was to fail, right? So yeah.
Maybe walk through that flow. From what I understand, or what I'm assuming, is that IPA has sort of a library of antibodies prepared that they've discerned through traditional lab methods. Now you're taking over from their data and seeing the applicability for the latest coronavirus, and then that goes back to the lab for validation. Walk us through that.
Yeah, sure. Yeah, so where we're at today, IPA is working with us together, and we're leveraging open research that's widely available, working with those sequences that were antibodies that target SARS-CoV-1, and seeing if we can develop a cross-reactive antibody that has good binding affinity to the latest strain of coronavirus. In several weeks from now, when that expression is done, they should have those antibodies sequenced. They can then provide us those sequences, and we can try to improve improve in many contexts of the word, the antibody sequences that they provide us in a second round. So it's kind of a two-phased project. One is with known sequences that have been published against the original SARS and try to make them cross-reactive. And then the second part, which is a lot more salient for us, is going to be to work with the actual proprietary sequences that IPA comes up with in their lab, and see if we can improve those features with our models by docking those antibodies against the target, and characterizing variations of them.
Good stuff. We're on the race.
Yeah.
Yeah, and I think you really are doing social good here, and I know that's part of your mission,
Mm.
and we applaud you for that. I know this is very stressful times, and Don and I just this morning we released a little note saying, "Hey, we're still here. We didn't publish last week. We published most weeks, and it's really changed our life suddenly."
Mm.
So the pressure's on for you, but we're supportive and we appreciate what you're doing here, both from a practitioner side and as fellow human beings. So thank
Thanks.
you. And thank you for being on our podcast. We really enjoyed having you.
And what help do you need from our listeners?
Do you want to do you need any help, or do you want to, do you want people to leave a way to contact you or anything like that, or to reach out?
Yeah, yeah. I mean, I think on our website there's a contact form if anyone has any questions or they have any suggestions or any interest at all. I think it's always welcome. Anyone's welcome to check out our website. It's EVQLV.com. So it's pronounced evolve, but the reason it's EVQLV, it's a common like amino acid kind of like a motif that's common to antibodies on their end
Got
terminals.
it. Clever,
Yeah.
clever. Love it. Thank you so much, Brett.
No,
Really
thank you
enjoyed
guys.
it.
Yes, thank you very
Thank
much.
you.
And some optimism for our listeners.